Hi, everyone. Thank you for the introduction. I'm Mackenzie, and today I'm going to be presenting about colorectal cancer and the microbiome. First, I'd like to discuss colorectal cancer. Colorectal cancer starts off as a polyp. Usually, serrated polyps, which are flat and harder to biopsy, are more likely to become cancerous. Colorectal cancer is one of the most common cancers, along with breast, prostate, and lung cancer. Incidence in people older than 55 years old is currently decreasing, likely due to increased screening, but those younger have rates increasing by 1 to 2% per year since the 1990s. Currently, it's a top leading cause of cancer deaths within the US. It's third for men and fourth for women. Next, let's discuss the microbiome. Studies have shown that risks of colorectal cancer includes the microbiome. The carcinogenic gut microbiome includes Bacteroides fragilis, Escherichia coli, and Fusobacterium nucleatum. Interestingly, however, these do not account for the majority of colorectal cases. There's a wide range of variation between studies, which suggests the Anna Karenina principle, that is, disease-associated dysbiosis are more varied than health-associated microbiomes. Now let's get into the study. This study compared the gut microbiome of colorectal cancer to health-associated gut microbiomes. They predicted the gut microbiome associations are more varied than are currently recognized. They identified protein-coding co-abundant genes, referred to as CAGs, clusters that may or may not be currently linked to colorectal cancer and precancerous adenomas. CAGs are part of microbial genes that have similar abundance within multiple samples, which suggests genetic elements that are shared by multiple strains. This is so that grouping within the same species is possible and that strain-to-strain variation doesn't limit associations, which is a common limitation of the traditional taxonomy-based analysis. This study used meta-analysis of surveys with ultra-deep shotgun metagenomic sequencing data in 5 countries across North America, Europe, and Asia. Nearly 1,000 people were analyzed in total. Colorectal association scores were generated where metagenome and colorectal cancer association was based on weight of the gene and its abundance. Metagenome colorectal cancer association scores were validated with human participant fecal samples. Each participant was divided into cohorts and provided 1 to 2 samples each. There were 3 cohorts total: colorectal cancer, in which samples were collected prior to treatment; history of advanced adenoma, which included a history of advanced adenoma with resection; and the final being the healthy cohort. Mouse experiments were performed to evaluate bacterial association with tumorigenesis and colonic gene expression. They were fed moderately high-fat diet, given antibiotics to be pathogen-free, and then human fecal microbiota was transplanted. Histological analysis after collection gave each tissue an inflammation score based on severity, extent, and mucosal architecture. Next, let's get into the results of the study. Over 2,300 microbial CAGs were identified that were significantly correlated with either colorectal cancer or health. They had varying abundance. Colorectal cancer-associated microbes include Bacteroides, Proteobacteria, and Fusobacteria. And the health-associated microbes included Actinobacteria and Firmicutes phyla. Of those identified, many included CAGs that were not previously associated with colorectal cancer, which is what makes the study unique in that they used CAGs in metagenomic sequencing instead of the traditional taxonomy classification approach to give them even more detailed data. They found that nearly 40% of gut microbiota might be carcinogenic, chemoprotective, or otherwise affected by colorectal cancer. More specifically, over 10% of CAGs were significantly correlated in colorectal cancer, and 12% were significantly correlated with health. Comparing this to the traditional taxonomy analysis, only 25 species have previously been correlated with colorectal cancer or health. Because of this new finding, I wanted to include some of the bacteria and their strains for completeness sake, which were previously not identified as cancer drivers. This list is based on colorectal cancer association scores. Figure A on the left shows the colorectal cancer cohort had more colorectal cancer associated CAGs when compared to the healthy cohort, which had more health-associated CAGs, and the log ratio of this data was greater in colorectal cancer microbiomes. Figure C on the right shows the number of health-associated genes was inversely proportional to the colorectal cancer-associated genes. This was also seen with the individual bacterial genomes, which was determined by comparing their metagenomic analysis to the NCBI reference sequence database. Colorectal cancer-associated CAGs were more likely to occur along with other colorectal-associated CAGs. I thought this figure they made was a great representation showing the significant correlations in the most common colorectal cancer-associated CAGs and the health-associated CAGs. And the thickness of these lines shows the correlation coefficients. Advanced adenoma microbiomes had significantly higher CAG-based colorectal cancer association scores, and those scores were significantly higher in the colorectal cancer cohort than in the adenoma cohort. There were a wide range of associations identified. The adenoma cohort had a bimodal distribution, and this heat map shows the top 10 colorectal cancer-associated CAGs and the health-associated CAGs. These findings further suggest that there's a high level of variation in the microbiome associated with colorectal cancer risk. Using both colonized germ-free mice with an APC mutation and IL-10 deficient mice, colorectal cancer-associated microbiota induced significantly greater tumor burden than health-associated microbiota. The tumor burden was high, but inflammation wasn't, which suggested that senescence might be a big driver. Microbiome-induced senescence was then investigated, which showed no growth-promoting products were secreted by the colorectal cancer-associated microbiota, suggesting that these bacteria have direct growth-promoting effects. But colorectal cancer-associated microbiome may be indirectly inducing tumorigenesis, as there were higher expression of features of senescence and inflammation, which are known colorectal cancer drivers. Colorectal cancer-associated microbiota transplanted mice had a greater proportion of senescent cells, including stromal cells, in particular fibroblasts, and macrophages. The microbiome-induced senescence creates a microenvironment that promotes epithelial growth and creates an opportunity for cancer to form. And finally, we're on to the discussion. They determined that nearly 40% of gut bacteria is associated with colorectal cancer or with health, and that senescence induction from the microbiome plays a role in precancerous effects. Another report from Japan previously linked two Porphyromonas species to gut senescence, and this current study allows them to generalize their findings. They further solidified that the microbiome induces fibroblast senescence and promotes epithelial growth. This leads us to senescence that is microbiome-induced and can be considered a precancerous state. These precancerous changes influenced by the microbiome may precede adenoma development, which leads us into how this data can then be applied clinically in the future. This CAG-based approach can provide a potentially targeted colorectal cancer prevention strategy. Future studies are needed to investigate which microbes are the strongest senescence inducers, and we can also evaluate the possibility of more accurate, non-invasive colorectal cancer screening in which microbiome CAG-based detection of precancerous lesions is performed with just a simple fecal sample. In conclusion, the microbiome is linked with colorectal cancer and might serve as biomarkers and targeted preventative therapy in the future. This study showed that CAG-based screening is needed to have more accurate colorectal cancer screenings. An important example of this is Faecalibacterium prausnitzii, which is known as an anti-inflammatory and health-promoting, but encoded CAGs with both colorectal cancer and health links. This goes to show that subspecies and strain identification can help identify which should be labeled as colorectal cancer associated and potential targets of precancerous therapy. And here are my resources, including the study. And what questions does anyone have?